Hello, my name is Mr. Chipman, and this is 5.3 Mendelian Genetics, Unit 5, Heredity, AP Bio. I teach AP Bio at Murray High School in Murray, Kentucky. I also teach honors biology. Taught several other classes over the years, making videos for AP Environmental. Stay busy. It's a good thing. Staying busy is a good thing. Let's talk about Mendelian genetics. It's called that because of this man, Gregor Mendel. Gregor Mendel was an Austrian monk, and he discovered these principles while he was working in the monastery, working with pea plants and bees in particular. Pea plants are what we typically use because we can understand these traits, things like green and yellow and wrinkled and round and all of those things. And he came up with these principles of genetics that we largely use today. I'm going to talk about in 5.4 how there are some instances in which these principles don't really work. We call that non-Mendelian genetics, shockingly. Mendel didn't know anything about DNA and that sort of thing either. And so, so a lot of these principles still work for us today, and we still talk about it quite a bit. So let's do that. A couple things we need to know first. Concepts of genotype and phenotype. Genotype and phenotype are words that we'll use a lot in this unit, and really throughout the year. We'll talk about phenotypes when we talk about evolution and ecology in particular. Genotype is a genetic code. We'll use terms like dominant and recessive, homozygous, heterozygous, in order to, do I have words for those or places for those? No, we'll show those later, so I'll make sure that we understand those terms. But when it comes to genotype, I always teach that it's what you can't see. When you look at a person, you don't see homozygous dominant or heterozygous, right? It's in the genes. Phenotype is the physical expression, right? Or physical characteristics, if you want to think of it that way. Brown hair, blue eyes, these are the things that we see, right? And so a phenotype is something you can look across the room and you can see that person is tall, right? That is a phenotype. That person is heterozygous tall. You don't see heterozygous, right? You just see tall, short, blue, brown, whatever. You get the idea. Phenotype, genotype. Very important words. If you get those mixed up, it's really hard to do anything in Mendelian genetics. So let's talk about the law of dominance real quick. The law of dominance is the idea that some alleles are dominant. Alleles, big word. Just used a word I haven't really talked about yet. I talked about it a little bit in 5.12, maybe. So alleles are versions of genes. So you look at these cherries. Two cherry trees came together to make these potential offspring that you see here. And each cherry tree donated a gamete, right? A haploid, fertilization, diploid, zygote, all of those things. And on the left, you see two dominant alleles. One cherry tree donated a dominant allele. The other cherry tree donated a dominant allele. And so they're the same. So we use the prefix homo for same and same zygote, right? And so meaning that the zygote that came together, fertilization, haploid gamete, diploid zygote, is a homozygous, is what we say. Homozygous dominant, because it's dominant. There's also homozygous recessive, which is what you see with the yellow cherries there. Two alleles or two gametes that had the recessive allele came together, and you get a homozygous recessive individual. In this case, red is dominant over yellow in cherries. Who knew? And then the other one is heterozygous. No need to say heterozygous dominant or recessive because it is heterozygous. And the dominant expresses itself over the recessive, which is the law of dominance. The law of dominance states that there are dominant and recessive alleles, and the dominant allele always expresses itself. Can the recessive allele express itself? Sure, but there can't be any dominant alleles around. The law of segregation. The law of segregation says that each allele has an equal chance of being inherited. What do we mean by that? So here we have some sort of condition, right? And the condition is recessive, and neither one of these individuals have that allele, right? Or they have the allele, neither one of them have the condition, unaffected. But they're called carriers. Carriers are a term you'll see a lot because they carry the recessive allele. But why don't they express it, or why don't they express it halfway? They don't express it because of the rule of dominance, right? Dominant expresses itself, so in this case, the dominant allele is unaffected or normal or whatever you want to say, but they're both carriers. And so dad here has an equal chance, 50-50 chance of donating a big R or a little r to its offspring. And you see that with this little thing here. But it also has a 50% chance of donating little r and little r, or little r. Why are we doing 4 here? Why 2 and 2? Remember, meiosis, 4 gametes, right? All that stuff. Okay. And so mom, on the other hand, has the same thing. Big R, big R. So, and this shows the probability. There's a chance that you'll have 2 big R's come together. There's a chance that you'll have a bigger chance that you'll have big R, little r come together, and a chance that little r will come together. And so that is called the law of segregation. Even though the recessive allele doesn't express itself if there's a dominant around, the recessive and the dominant alleles segregate from one another. When do they do that? They do this during meiosis 1, right, when homologous pairs separate. So this goes back to meiosis. Tempted to pull in another slide, but I'm not going to do that. This goes back to meiosis when you have homologous pairs. Mom gave you a big A, dad gave you a little a. Now you're making gametes, and big A and little a are going to separate during meiosis 1, right? So very important to link those two things together. And then the law of independent assortment, which we've already talked about. But it has a little more legs now, so let's look at it. Zoom in. So this individual has alleles for whatever trait A is, right? They have one of each, dominant recessive, so they're heterozygous for A. They're also heterozygous for B. A and B are located on separate chromosomes, and those separate chromosomes independently assort themselves, right? So A and big A and little a are going to segregate during meiosis 1, right? That's what we talked about, which is up here, right? Big A, little a, segregate meiosis 1. They say metaphase, I say anaphase, whatever, doesn't matter. It's all the same sort of idea. And big B and little b segregate during meiosis 1 as well. But when does big A and big A separate? Well, that's meiosis 2. And so you see that A's have an equal chance of being inherited. What about B? What about big B? Does it go with big A, or does it go with little b or little a? Well, they can do either. And so this shows those combinations. And so they independently assort themselves. And so if you have an individual that is heterozygous for both B and A, then they have multiple alleles that they can offer, right? And those combinations become a lot more complicated when you do that. This brings us to the concept of fertilization. We can demonstrate fertilization by showing something like a Punnett square, right? Because we talked about the random fertilization, and we use Punnett squares to show the probability of one allele from one person coming from or combining with another allele with another person. And very simple concept here. Just kind of breeze over this. So if you have a homozygous dominant, homozygous recessive, what is the chance that homozygous dominant will donate a dominant allele? 100%. 100% little p as well, and so 100% of the population is going to be, or the offspring are going to be big P, little p. Get the idea. So let's look at these laws of probability when it comes to talking about things like this: fertilization, chances, and independent assortment, and segregation. All of this sort of builds to this idea of, okay, so then if we have these two parents and we want to predict what is the likelihood that this offspring will be born between them, right? How do we do that without setting up some big Punnett square and doing all the things? Like if you had me in honors, I taught you how to do all that, and it was slow and it's tedious and all that kind of thing. And so let's talk about how to do this. Well, we're going to use these laws of probability that we see over here. You probably had these in a math class maybe, and the mutually exclusive and independent and all that kind of stuff. You know, it's a lot of stuff going on. So let's look at here. So if we have these parents, and what is the chance of this offspring happening, we can use these laws of probability in order to do that. And so let's look at the A's independently first. So if those A's, if those two individuals with those particular genotypes for A, whatever A is, combine, what is the likelihood that they will have an offspring that is little a, little a? Well, I know that there's a half chance that little a will be donated by this individual, right? And there's a half chance that little a will be donated by this individual. And so there is then a half times 1/2 is little a, so that is 1/4. We're going to just write that here, right? So 1/4 for A. What about B? Well, what's the chance that the big B, little b, is going to give away a big B or a little b? Well, that is going to be, it can be a big B one time or it can be a little b the next time. It's kind of like a choice, right? And so if you were to do a Punnett square between big B, and big B, little b, sometimes it's going to be big B, little b, but sometimes it's going to be little b, big B, right? And so 1/4 of the time it's going to be one big B, and another 1/4 of the time it's going to be another big B, little b. And so you add those two things together, right? So 1/4 plus 1/4 is 1/2. So what is the chance that we will have big A, little a, and big B, little b? Anytime there's an and, you multiply. It will be a, that's an equal sign. It will be a 1/8. I love how that's just, let me solve this for you. Or 0.125. And so, yeah, that's how you do those. You can do this with, I mean, you could just keep going out. You could just make it long. You could do this, right? Used to, you'd see problems like this on the exam. Not so much anymore. The big place where you're going to see this on the exam is going to be with probability with chi-square, right? And so just a couple of rules that you need to know. One of them that you will see quite a bit is the phenotype, expected phenotype ratio on a cross with two heterozygous, and it's going to be 9. I'll just go ahead and add this rule because I think it's important. 3, 3, 1. Whereas you have 9 that are homozygous, are both dominant phenotypes, 3 that are one dominant, one recessive, and 3 that are the other dominant and the other recessive, and then 1 out of those offspring that are going to be both recessive. Again, those are percentages, but with 16 total possibilities, right? So 9, 3, 3, 1. That is the thing that I continue to see. Otherwise, it's just a probability problem. So let's look at the different kinds of crosses. A monohybrid cross. This is a cross between just looking at one particular trait, like blue versus white here, whereas blue is dominant, white is recessive, and that's a monohybrid cross, right? You can look at the probabilities there. A dihybrid cross is when you have multiple, or not multiple, just 2 different traits. Here we have yellow and green, and then round and wrinkled. And so this would be an example of independent assortment, as long as they're on different chromosomes. We'll look at same chromosomes in another lecture. And so if yellow and green are one allele and round wrinkled another allele on a separate chromosome, then you can expect that they would independently assort themselves. And you have all these different possibilities with that 9, 3, 3, 1. 9 is both dominant. 3 is dominant round, recessive green, recessive wrinkled, dominant yellow, and both recessive, right? And again, for any one of these, if you have this, or if both of them are heterozygous for any one of these, you could solve the percentage by just doing the probability math that we did up here. So let's look at this idea of autosomal inheritance. Autosomal inheritance has to do with what we've been talking about, right? An autosomal chromosome is a chromosome that is not a sex chromosome, right? And it will typically follow pretty normal patterns. Dominant inheritance is, dominant is always going to express itself. So if you look over here, you can see that this particular parent has this, whatever blue is, right? They have blue because it's dominant, but they're heterozygous. So their partner here is homozygous recessive, and so there's a chance that they'll have both kinds of children. Over here, if it's a recessive trait that's being tracked, both of these parents are carriers. Carrier, again, doesn't mean they express anything, just means they carry the recessive allele. And so they have a chance to have an individual who is homozygous recessive for both traits or for both alleles, or homozygous dominant, right? And so they could be either one. There's some other kinds of inheritance we're going to look at. X-linked inheritance is this idea of inheritance that is only found on sex chromosomes. Why is this a big deal? Well, it's a big deal because males only have one X chromosome, right? And so they either are dominant or recessive, whereas females can be heterozygous, homozygous dominant, homozygous recessive. And that is going to create some different kinds of patterns, right? Because not only are you inheriting dominant recessive blue-white here, but you're also inheriting XY or XX, right? Dad gives away an X or a Y, mom gives away an X or an X, right? And so a couple of rules that are helpful when dealing with sex-linked or X-linked conditions. The son always gets the X chromosome from mom, right, every time. And so because the Y always has to come from dad, mom doesn't have a Y to give because mom's mom. And so sons always inherit the X chromosome from their mother. And so if the mom has two of the same X chromosome, then the sons are always going to have that condition, right? Whether it's dominant or recessive. Fathers give an X to their daughters, right? And they only have one X to give. So there's a couple of things that are helpful when you're looking at those. This pedigree, I mentioned pedigree, we're getting ready to talk about pedigrees, but these lines of inheritance are going to be something you'll have to be able to solve between when you're looking at different kinds of problems, which brings pedigrees. And so pedigrees, you'll see these quite a bit, these types of pedigrees where you'll see a pedigree, maybe 3 generations, and it'll say like, what type of inheritance pattern do you see here? And you sort of have to use process of elimination to decide that. And so in this particular case, right, autosomal dominant, I'm assuming that in this particular picture, you have, the pink equals homozygous recessive, and then blue equals the dominant allele, right? And so then in a dominant one, it's easy to see. Dominant's always going to express itself, right? Maybe blue is homozygous dominant in this case. Yeah, that's what's going on, and white is heterozygous. In a autosomal recessive pedigree, look, both parents don't have the trait, but one of their children does. How's that possible? Well, because they both are able to give that away. There's a 0.25% chance that they'll have a child with a homozygous recessive allele. With X-linked recessive, right, again, dad gives the X only has one X and gives it to their daughters every time. And so if it's dominant, there's no chance that their daughters are going to have that trait. There's a chance that the sons might have it, though, right? Because mom gives away 2 X's, but in this case, she's heterozygous. You can kind of figure that out. X-linked dominant trait, if mom has it or has the dominant trait, there's a chance that she'll give it to her sons. Dad doesn't have it in this case, so he can't give it to his daughters. You get the idea. Right, Y-linked. It's only where you might see a Y-linked. We're going to look at another one that you'll see, mitochondrial. I think we're going to look at that in 5.4. So let's look at a practice problem. Breeder wants to cross 2 heterozygous tall plants, which the following would most likely occur if a dominant tall allele mutated and lost its function, making it recessive instead. Interesting question. If the dominant allele mutated, so it doesn't change the allele, right? Making it recessive. Let's look at some of these answers. This is going to be difficult. I get this question a lot from students. This will be interesting. Phenotype ratio of offspring would remain the same because allele function does not affect inheritance. That is not true. All offspring would display the tall phenotype as T is still present in both parents. No, it doesn't, because making it recessive instead is mutation. The expected phenotypic ratio of 3:1 would shift to 1:3 with fewer tall. Again, no, it's not going to happen because it makes it recessive. So D is the correct answer because A, B, C, and wrong. But let's look and see why D is the correct answer. All offspring would display the short phenotype correct, making it short That was not hard as I thought it was going to be, but still you had to kind of think through them and look at them. I hope this was helpful. If you have questions for me, please ask me down in the comments. I know that this can be pretty difficult for some students, right? Because it's a lot of math, it's a lot more logical thinking. For some of you, you're like, man, this is a breeze. For others of you, are like, wow, this really sucks. Unit 5 is kind of one of those units that is very polarizing in that way. So if you have questions, leave them in the comments down below. Thank you for watching.